Every second, an invisible clock ticks within each of our cells, dictating the pace of life and the inevitability of aging. This isn't a metaphor, it's a measurable biological reality governed by structures called telomeres. Located at the ends of our chromosomes, telomeres act as protective caps, safeguarding our genetic information. But with each cell division, they shorten, like a fuse burning down. When they become too short, cells lose their ability to divide properly, entering a state of senescence or programmed cell death. This process is intimately linked to the onset of age-related diseases. For decades, the quest to slow this biological clock remained elusive. However, a growing body of research is pointing towards a lifestyle intervention that doesn't involve complex drugs or invasive procedures: intermittent fasting. Far from a fad diet, intermittent fasting, or IF, is an eating pattern that cycles between periods of eating and voluntary fasting. Its proponents claim benefits far beyond simple weight loss, suggesting a profound impact on cellular health, metabolism, and even the very mechanisms that govern our aging process. The question is, how does this ancient practice intersect with our modern understanding of cellular longevity? The central hypothesis linking intermittent fasting to telomere health is compelling. While chronic inflammation and oxidative stress are known accelerators of telomere shortening, IF appears to mitigate both. By inducing a metabolic shift from glucose burning to fat burning, and by prompting cellular repair processes, fasting creates an environment less conducive to cellular damage. This isn't merely about slowing the rate of shortening. Some studies suggest that IF might even promote the activity of telomerase, the enzyme responsible for rebuilding telomeres. This implies a potential for active maintenance, not just passive protection. One of the most robust scientific arguments for IF's impact on telomeres lies in its ability to reduce oxidative stress. Free radicals, byproducts of metabolism and environmental factors, can damage DNA, proteins, and lipids, including telomeres. Studies on fasting regimens have consistently shown a decrease in markers of oxidative damage. For instance, research published in the journal Cell Metabolism indicated a significant reduction in lipid peroxidation, a key indicator of oxidative stress, after just a few weeks of fasting protocols. This directly translates to less collateral damage on those crucial telomeric caps. Beyond stress reduction, intermittent fasting supercharges a critical cellular process called autophagy, literally meaning self-eating. This is the cell's sophisticated recycling program, where damaged components, misfolded proteins, and dysfunctional organelles are broken down and removed. Active autophagy is essential for maintaining cellular health and preventing the accumulation of cellular debris that can accelerate aging and contribute to telomere attrition. By initiating periods of nutrient deprivation, IF triggers this cleansing mechanism, essentially spring-cleaning our cells and potentially protecting telomeres from a buildup of harmful byproducts. The scientific promise of interventions like intermittent fasting and telomere maintenance isn't just a topic for academic journals, it's increasingly a driving force in the global economy. Venture capital and institutional investors are pouring billions into the longevity sector, seeing the potential for substantial returns on investment in therapies and technologies that extend healthy human lifespan. The market for anti-aging and regenerative medicine is projected to grow exponentially, with public companies specializing in biotech and health innovation showing impressive performance on major exchanges. This financial dynamism underscores the profound belief in the scientific breakthroughs in cellular longevity. While intermittent fasting shows great promise, it's crucial to compare it with other well-studied longevity interventions, such as continuous caloric restriction, CR. CR involves consistently reducing daily calorie intake by 20 to 40% without malnutrition. Both IF and CR have demonstrated benefits in animal models for extending lifespan and healthspan, often through similar metabolic pathways. However, adherence to CR can be challenging long-term. IF offers a potentially more sustainable approach for many, achieving some of the same cellular benefits without the constant psychological burden of daily deprivation. The key distinction often lies in the feasibility of long-term compliance and quality of life. Despite compelling evidence, the science of intermittent fasting and telomere length is not without its nuances. Research results can vary widely based on the specific IF protocol, individual genetics, lifestyle factors, and duration of the intervention. More long-term, large-scale human trials are needed to solidify these findings and understand optimal strategies for different populations. Not everyone responds to IF in the same way, and what benefits one person might not be suitable for another. The ultimate goal is to move towards personalized longevity strategies, where interventions like IF can be tailored to an individual's unique biological makeup and health goals. The journey into understanding the intricate relationship between intermittent fasting and telomere length is just beginning. What we've uncovered points to a powerful, accessible tool for potentially extending our health span, allowing us to live not just longer, but healthier, more vibrant lives. As research progresses and our understanding deepens, IF may become a cornerstone of personalized anti-aging strategies. The convergence of ancient wisdom and cutting-edge science offers an exciting glimpse into a future where we can actively invest in our cellular health, pushing the boundaries of what's possible for human longevity. This is more than health, it's a paradigm shift.